Shackey has focused his research on aspects of cardiovascular prevention, such as omega-3 fatty acids, aspirin, ACE inhibitors, diabetes, and others. Together with a colleague, he invented and defined the omega-3 index, a standardized method for erythrocyte fatty acid analysis. Professor von Shackey, please, the stage is yours.
Thank you very much for your kind introduction. I go right on with my presentation. This is my conflicts of interest. When we are talking about fatty acids, basically we are talking about the cell membrane. And the cell membrane is important for the function of proteins like signal transduction molecules or ion channels. And all these proteins interact with the fatty acids around them, and the fatty acids around them have a modulating effect on the function of all these proteins. And each and every cell has a distinct idea what fatty acids it wants to have in the cell membrane. Of course, nature is, let's say, generous, and the cell gets by with an suboptimal composition of fatty acids, but it functions optimal with a specific fatty acid composition. Nutritionists told us that we can convert plant-derived alpha-linolenic acid to the marine icosapentaenoic acid, and this one to DHA, which we have in the brain, for instance, and which we need to build our brain with and to maintain our brain with. But these data are true for rodents and not for humans. Humans poorly convert alpha-linolenic acid to EPA and do not, basically not, convert EPA to DHA, meaning we need DHA, we need a supply with DHA to build and maintain our brain and to build and maintain the cell membranes of each and every other cell in our body. There are many honorable scientific associations like the American Heart Association or the World Health Organization who recommend daily intake of omega-3s. But if you look at the dose recommended, that varies from 250 milligram to 1 gram, 2 to 4 grams. So there is an uncertainty about how to dose the EPA and DHA, the marine omega-3 fatty acids. If you give such a recommendation, you assume that what you put into your mouth, because that's what is recommended, also shows up in your body, meaning is being taken up by your body. But this is not guaranteed. Omega-3s are very well absorbed with a high-fat meal because they need the digestion of fat. They are fats. And it doesn't really happen with a low-fat meal. And this sounds very simple, but actually is one of the reasons why we have so many neutral intervention trials, because those intervention trials that were run by clinical research organizations, and there were many of them, they always recommend to have a capsule or two of omega-3 fatty acids with breakfast, and in many countries, breakfast is a low-fat meal. Emulsion is important. Something we call matrix effects are important, and there is a large variability from person to person in how bioavailable omega-3 fatty acids are. In other words, intake doesn't have much to do with uptake. Individual bioavailability is not predictable because of that factor 13. and you need to individualize dosing of omega-3s. So these recommendations, they mean well, but on an individual level, they're basically useless. And besides, cardiologists and everybody else knows that what goes through our arteries is not components of food, it's blood. That's why we chose to measure in blood, and we invented the omega-3 index. The seminal publication was in 2004. It's a biomarker for fatty acid composition, and the red cell correlates with each and every other tissue that we looked at in terms of fatty acid composition. The omega-3 index is measured in red cells, and they have a low biologic variability in terms of fatty acids, meaning we are basically looking at something equivalent to a hemoglobin A1C and not at blood sugar. We have a standardized and validated method for 26 fatty acids, and we start learning more and more about each and every fatty acid that's present in the cell membrane. And we have now 360 publications based on our method, which is the largest database for any fatty acid analytical method. And the trick is the low biological variability. Whole blood is half red cells and half plasma, larger biological variability. And in plasma or in plasma phospholipids, the biological variability is much larger. Now, how does that play out in a meta-analysis? If you analyze whole plasma, then you cannot be sure that you have a good signal to noise ratio. And as you see here, 3 of the 4 studies, they cross the line of 1, meaning they didn't show any differences. On the other hand, up here, whenever red blood cells were analyzed, you don't get to cross the line of ineffectiveness, and you have a good signal to noise ratio. In this case, it was total mortality and EPA and DHA in blood per interquartile range. Same thing for later development of type 2 diabetes. Analyzing adipose tissue is not informative as it is true for triglycerides, cholesterol esters, total plasma. And if you want to see something, you better analyze red blood cells. So the informative value of EPA and DHA is best in red cells, worse in whole blood, and you don't want to really analyze all the other fatty acid compartments that are listed here. If you compare the red cell in terms of fatty acids, you will find that it is representative for heart tissue, atrial and myocardial muscle, breast, peripheral muscle, mucosa, placenta, that has been done in humans, and in a lot of other tissues that have been analyzed in experimental animals. And again, the correlation with intake is poor. Now, analyzing red cells sort of became fashionable in clinical labs, and we sent 2 samples out to other clinical labs, one which we call control high, and one which we call control low. And the results we got back from the other labs were all over the map. And they overlapped high controls and low controls between the other labs. So it is absolutely necessary from my point of view to use a standardized analysis and to standardize together with the other labs to get the same results. Now, if you analyze a lot of samples with our method, you get to see what you expect for a biomarker that is a statistically normal distribution. What I did not expect to see was that there was not a single human being without an omega-3 index. So these omega-3s are something humans cannot live without. And you don't get to see levels higher than 20, which, by the way, is also true for dolphins, and who eat fish all day, and yet they don't have an omega-3 index above 20. So this seems to be tightly regulated, and it tells me every human being has an omega-3 index. EPA and DHA are vital, and we defined the target range as 8 to 11%. Now, some people, like vegans, they don't eat sources of omega-3 fatty acids, EPA and DHA, and of course they have low levels. In some countries, like in South Korea, people eat a lot of fish, and 80% of them have an omega-3 index in the target range. And if you use this kind of data and you draw a world map, you get to see the countries that have low levels, and Israel certainly is among them. Other countries like Japan or Norway or South Korea have high levels, and they are in the target range. So let me sum this up. The fatty acid composition of the cell membrane modulates cell function. Many scientific societies recommend intake of EPA and DHA for whatever reason. In humans, plant-derived alpha-linolenic acid is barely metabolized to EPA, and EPA not to DHA, but we need DHA to build and maintain the brain. Intake and uptake don't have much to do with each other, but for clinical events, cell levels are decisive. We have this standardized determination of level. In Western countries and in Israel, levels are low. And let me tell you what the levels mean. We have a number of epidemiologic studies on that. One of them was conducted in Germany. This is 3,500 individuals in the Ludwigshafen area. We determined the omega-3 index at the beginning and then waited for 10 years and related the omega-3 index to the total mortality occurring in about 1/3 of these individuals. And as you can see, with an omega-3 index of above 8, the probability to die in a 10-year time frame is only 2/3 of the probability to die with an omega-3 index below 4. This is a powerful risk factor for total mortality. We have identical results from the US, from the Women's Health Initiative memory study. Same picture, same figures. And we also have very similar results from the Framingham study, demonstrating that with a low omega-3 index, total mortality is much higher than with a high omega-3 index. And again, the figure tells you it's 2/3. If you compare the relevance of the omega-3 index to cholesterol, an established risk factor, then cholesterol is not informative. More data from Framingham. We tried to assess the relevance of the omega-3 index. In a group of 65-year-olds, those who had a low omega-3 index and did smoke, 50% died within a 10-year time frame. If you either have a low omega-3 index or you smoke, then total mortality was 25%. And if you don't smoke and have an omega-3 index in the target range, total mortality is 12.5%. These, to me at least, are fairly large differences because I happen to be 66 years old and I want to be on that line and not on this line. Now, how about cardiovascular events? Again, data from Framingham, and they showed you all the same relative data. 2/3 of events with an omega-3 index in the high range as compared to an omega-3 index in the low range. And for stroke, it's only half. Again, cholesterol was not relevant: total cardiovascular disease, total coronary artery disease, cardiovascular mortality, and overall mortality. The fact that clinical events, important clinical events, and maybe also not so important clinical events correlate with the omega-3 index is also true for intervention trials. Increasing the omega-3 index improves a lot of surrogate parameters like heart rate variability, blood pressure, reduces inflammatory biomarkers, triglycerides, and changes the composition of LDL towards a less atherogenic composition. And omega-3s do reduce blood pressure. They reduce pro-inflammatory cytokines. This is a meta-analysis of, for instance, here 50 trials on blood pressure. They reduce heart rate. They reduce the interaction of blood with endothelium and improve endothelial function. No question. And also intermediary parameters like coronary lesions and left ventricular remodeling after myocardial infarction are improved. And in terms of coronary lesions, they work together with statins, as you can see here, and improve the effects of statins and vice versa. And the remodeling of the left ventricular infarction after left ventricular infarction is improved. And what's important here is that, again, the parameters that are improved correlate with the improvement of the omega-3 index. Now, you've seen all these meta-analyses maybe that didn't show much in past years, but the more recent meta-analyses show more positive effects. This is a meta-analysis from last year. Overall, a positive effect of omega-3s. And one of the trials that's in here was the Alpha Omega trial. It was run in the Netherlands. And in 2016, I gave a presentation to this group. And then they went back to their freezers and they analyzed what they had. They had some plasma left, and they realized that, yes, there is a relation between events and omega-3s, but it's the baseline value and not the intervention that they did. Why are there so many neutral trials? I was present when the ASCENT trial was presented, and this is a group from Oxford, and they said, well, we know now that omega-3s don't work. And our sister lab in the US had analyzed a number of samples from that trial, and it turns out that in the Verum group, on average, the individuals were recruited with a high baseline omega-3 index. And if there is no room for improvement, there cannot be any effect of omega-3 fatty acids. Ignoring baseline values, and baseline values don't have much to do with how much fish people eat, is a reason for many neutral trial results. A dose that was frequently used was slightly below 1 gram. And if you take the baseline values for the placebo group and say, okay, that's the Verum group, you see that the levels in the Verum group are not higher than in the placebo group. And if you don't separate the levels, you won't get to see any effect. Of course, you can use a lot of stuff and then separate levels and see an effect. The meta-analyses tell us that longer times and higher doses are more effective than shorter times and lower doses. And this has also been demonstrated in the REDUCE-IT trial, a 5-year trial with a high dose. Serum levels were measured, increased 6-fold, and there was a 25% reduction in the primary endpoint and in all other endpoints observed. For instance, also a 28% reduction in fatal or non-fatal stroke. This trial was then evaluated again from the perspective of the serum levels measured. And as you can see here, mid-high levels reduced clinical events. That's true for primary endpoint, that's for secondary endpoints, that's true for cardiovascular death, for total mortality, and so on and so forth. And if you compare these two evaluations of this trial, comparing verum versus placebo, you get a 25% reduction in the primary endpoint, but a 65% reduction in if you compare the levels reached. Much larger effect when using the perspective of the levels than when considering EPA and EDHA, or in this case only EPA, simply a drug. So ignoring baseline levels and the complex bioavailability caused many neutral results of these trials. To be positive, a trial must recruit a population with a deficit in baseline, must have room for improvement, and the levels in verum and placebo must differ. Nevertheless, the latest Cochrane meta-analysis was positive for cardiovascular mortality, for cardiovascular events, for coronary heart disease mortality, and for coronary heart disease events. So let me sum this up. In epidemiology, we have inverse correlations between clinical events and the omega-3 index. And we have correlations on surrogate endpoints, also with the omega-3 index, on intermediate endpoints. Of the intervention trials with clinical endpoints, many were neutral, and I think that's a methodologic problem. Nevertheless, the last Cochrane meta-analysis was positive for cardiovascular and coronary heart disease mortality. I conclude that a low omega-3 index is a risk factor for total mortality, myocardial infarctions, stroke, and other cardiovascular events. Now let me finish with considering safety and tolerability of omega-3 fatty acids. In the large and long trials with a high dose, this is where you would want to look for untoward effects, like the JELIS trial or the REDUCE-IT trial. You have the identical picture. You have bleeding events at 0.1% per year, 1 in 1,000, more in the verum group than in the placebo group. I think that's a phenomenon of overdosing, because if you give 4 grams a day, that's a lot of stuff, and 5 years is a long time. The other thing that came up recently was the observation that atrial fibrillation tends to occur in these trials that I talked about, like the REDUCE-IT trial. And it tends to occur more frequently with a higher dose than with a lower dose. The important thing is that there was no increase in stroke, which is the complication of atrial fibrillation that we fear the most. Consistent data from another meta-analysis. What was not discussed in these meta-analyses is that there is a U-shaped relation between the omega-3 index and onset of atrial fibrillation. And minimal risk for a new onset of atrial fibrillation comes with an omega-3 index in the target range. Lower levels come with an increased risk for atrial fibrillation, as do higher levels. Nevertheless, the European Food Safety Authority considers up to 5 grams a day as safe, and the US American Food and Drug Administration thinks up to 3 grams a day are safe. I'm a bit more cautious. I would say stick to the target range and certainly don't go above 16. And I would like to add the tolerability and bioavailability are maximal if taken with a main meal. I sum up again. We have a biomarker for a person's status in EPA and DHA, and that's the omega-3 index. The biomarker is important because intake doesn't have much to do with uptake. Many intervention trials focused on intake. Ignoring baseline levels and the complex bioavailability of omega-3s was responsible for many neutral trial results. The low omega-3 index is associated with elevated total mortality, sudden cardiac death, fatal and non-fatal myocardial infarction, stroke, congestive heart failure, and so on and so forth. And increasing the omega-3 index reduces mortality and reduces these clinical events. I could not talk about the other positive effects of an omega-3 index in the target range, but there are many with respect to brain, muscle in pregnancy, and so on and so forth. And I put it to you that EPA and DHA are safe and tolerable, especially when given under control of the omega-3 index. Thank you very much, and I'm happy to take questions.
From the public, is there a way for vegans to get EPA from vegan supplements or another way?
There are products on the market that come from algae, and many vegans take these products already. And in my country, there's even an official recommendations for vegans to take such a product because they need higher levels of omega-3s.
And Dr. Dana Tzfat has a question. Maybe you can ask him herself. Is there any correlation between omega-3 index and age?
There is a slight increase of the omega-3 index with age, and I'm not sure whether that is attrition of the susceptible, meaning that sort of the ones with the low omega-3 index are weeded out by fate, but I'm not sure whether that's true.
I think we'll finish with this and thank Professor von Shaekhi. Thank you for joining us.
Well, thank you very much. And again, I apologize for not being there. And I also apologize for not being able to give this presentation in Hebrew. I would love to speak Hebrew. It's a nice language.
Don't try my German. Auf Wiedersehen.
Thank you very much.
And bye-bye.
Bye-bye.
Our next speaker